everyone. We will be discussing natural selection in antibiotic-resistant bacteria. Alexander Fleming discovered penicillin, the first antibiotic, in 1929. Since then, other natural antibiotics have been discovered and many more have been synthesized in laboratories. So after Fleming discovered penicillin, the first antibiotic, other antibiotics have been discovered. Each bacterial type has a specific antibiotic. The use of antibiotics has increased dramatically, particularly over the last 20 years. This random use of antibiotics can be dangerous as it leads to the development of bacterial resistance to an antibiotic. As a result, the effect of the antibiotic will be lost or will be inefficient to kill the bacteria. Usually, mutations happen all the time in all living organisms. These mutations are due to an unorganized placement of the nitrogenous bases responsible for producing a specific protein. So any mutation will lead to a mutation in the protein being produced, and as a result, the characteristics of the individual will change. Mutations can be harmful, leading to a certain type of disease. They can be neutral, not causing any disease. However, another type of mutation can be beneficial. Like in our case, mutations in bacteria can allow it to become resistant to antibiotics, which will give the bacteria an advantage. In bacteria, a chance could give a bacterium resistance to an antibiotic. So the mutation will give the bacteria a chance to stay and resist antibiotics by natural selection. Here we can see a bacterial resistance to an antibiotic. Here we have a type of antibiotic on a bacteria. This is how the antibiotics are tested against a certain bacteria in the labs. They get a petri dish and they fill it with agar, which is nutritive. It allows bacteria to grow, and then they get a sample of the tested bacteria using a sterile loop and place it on the agar. Then they place the petri dish in the incubator for 48 hours at 73 degrees Celsius for the bacteria to grow and spread on the agar plate. Afterwards, they place a certain type of antibiotic and observe whether this antibiotic can kill the bacteria or not. Here we can see that this antibiotic is resisting the bacteria and killing it, since here we have an empty zone of bacteria, as you can see. So this type of antibiotic is killing the bacteria. However, here this antibiotic is not resisting the bacteria, since the bacteria developed a mutation in the gene which allowed it to resist this type of antibiotics for it to become an antibiotic-resistant bacteria. So due to environmental changes, this bacteria developed a mutation which gave it a chance to stay and increase. So this gene mutation will be passed on to the next generations, allowing the increase in the antibiotic-resistant bacteria. So in a situation where antibiotics are widely used, this new resistant bacterium has an advantage over non-resistant bacteria of the same type.